At first glance, building a synthetic cell might seem like something straight out of science fiction, a tiny living machine built from scratch in a lab. But in cutting-edge biophysics and synthetic biology, researchers are already doing just that. And one of the most promising tools in this pursuit? Microdroplets. These tiny cell-sized droplets of liquid are turning out to be powerful platforms for mimicking cellular compartments and reactions, giving scientists a way to simulate, probe, and even recreate lifelike behavior, one droplet at a time. Let's take a step back. Real biological cells are astonishingly complex. They're not just bags of fluid with some DNA floating inside. Cells are compartmentalized, crowded, dynamic systems, packed with membranes, organelles, enzymes, and molecular machines, all orchestrating a ballet of chemical reactions. Reproducing this level of organization from scratch is a colossal challenge, but that's where microdroplets come in. They offer a controllable, tunable microenvironment where lifelike processes can be studied one step at a time. So, what exactly are these microdroplets? They're tiny volumes, often just picoliters to nanoliters, of aqueous solution surrounded by oil or other immiscible liquids, sometimes enclosed by a lipid or polymer shell. In other words, they look and act a lot like cell membranes, especially when engineered with surfactants, lipids, or proteins. By using microfluidic techniques, researchers can create thousands or even millions of droplets, each with slightly different compositions, to act as individual experimental cells. One of the key applications of microdroplets is in mimicking compartmentalization. In real cells, compartmentalization allows different reactions to happen in different places, like how lysosomes digest waste while mitochondria produce energy. Microdroplets can be engineered to contain multiple inner compartments, or even to fuse and divide like cells do. Researchers can design droplets with membrane-like interfaces, phase-segmented interiors, or even programmable division cycles, mimicking everything from metabolic pathways to cell signaling events. You might ask, can we really simulate life in something so small and artificial? Surprisingly, yes. Within these droplets, scientists have successfully recreated gene expression, enzyme cascades, protein folding, and even minimal metabolic cycles. DNA templates can be transcribed into RNA, which is then translated into proteins, all within a droplet less than the width of a human hair. These systems can be controlled with external stimuli like light or temperature, and they provide a clean, isolated environment where each variable can be precisely adjusted. One particularly exciting area is bottom-up synthetic biology. Instead of modifying existing cells, top-down, researchers use microdroplets to build new ones from basic parts: membranes, nucleic acids, ribosomes, and metabolic enzymes. The goal is to create synthetic cells that are lifelike but not alive, able to sense, respond, adapt, and replicate chemical functions without the full machinery of natural biology. These droplet-based synthetic cells could have major applications. In medicine, they could serve as targeted delivery vehicles, releasing drugs in response to specific molecular signals. In biosensing, they could detect toxins or pathogens by producing fluorescent signals. In chemical synthesis, they could act as miniaturized, parallelized bioreactors, performing thousands of reactions simultaneously. But there are deeper scientific goals too. By building synthetic cells from the ground up, researchers can begin to answer some of the most fundamental questions in biology. What defines life? How does complexity emerge? Can we recreate evolution in a test tube? Microdroplets offer a platform for exploring these questions experimentally, not just theoretically. Of course, challenges remain. Real cells are not just bounded containers, they're dynamically regulated, capable of self-repair, adaptation, and evolution. Achieving that level of functionality in synthetic droplets requires advances in materials science, systems integration, and molecular programming. But progress is rapid, and with tools like CRISPR, synthetic genomes, and programmable liposomes, the frontier between life and machine is blurring fast. And perhaps that's the most mind-expanding part of all. In these droplets, we're not just observing biology, we're reconstructing its logic. Step by step, molecule by molecule, we are learning how to engineer lifelike behavior in simple, tunable systems. In the end, microdroplets are not just tools, they are tiny laboratories, synthetic cells in training, and portals into the very origin of life. They remind us that complexity can emerge from simplicity and that the line between chemistry and biology is, perhaps, not a line at all, but a bridge we're just beginning to cross. Microdroplets provide a powerful platform for the bottom-up construction of synthetic cells, serving as artificial compartments that mimic the spatial organization, confinement, and reaction dynamics of living cells. These droplets can be composed of aqueous phases enclosed by lipid, polymer, or protein shells, emulating biological membranes. The paradox lies in how such simple, minimalistic systems, just tiny drops of solution, can recreate lifelike behaviors, including gene expression, signaling, metabolism, and division, even in the absence of a full cellular genome or structure. Lipid-coated water-in-oil microdroplets have been shown to sustain transcription and translation, producing proteins inside droplet compartments without a living organism. It's like building a miniature kitchen inside a soap bubble where cooking happens independently, even though there's no chef, only the right ingredients and confined space. Cellular mimicry in synthetic biology refers to the recreation of core life functions, such as energy conversion, chemical processing, and selective permeability inside artificial compartments. Microdroplets act as cell-like reactors, confining reactants and enabling biochemical cascades with spatio-temporal control. The encapsulation enhances reaction rates, stabilizes fragile components, and allows for interactions akin to organelles. Despite lacking genetic or evolutionary complexity, these microenvironments are programmable, dynamic, and responsive, enabling researchers to define what constitutes a minimal cell. Researchers created droplet-based systems where metabolic enzymes convert substrates into products through coordinated reactions, mimicking metabolic pathways of living cells. It's like constructing a mini factory out of Lego blocks that autonomously assembles products just because the right modules are enclosed and connected inside. Droplet-based synthetic cells rely on key mechanisms such as phase separation, selective membrane permeability, diffusion-limited reactions, and programmable encapsulation. These features allow spatial segregation of reactions mimicking cytosol and organelles, maintenance of gradients, and autonomous replication of certain features such as lipid bilayer growth or vesicle division. In some systems, droplets can even communicate through chemical signaling, form colonies, or fuse like cells exchanging contents, blurring the line between chemistry and biology. Artificial protocells have been engineered to communicate via quorum sensing-like mechanisms, exchanging small molecules between neighboring droplets. It's like organizing a network of balloons that talk to each other using perfume. They diffuse signals that others smell and respond to in kind. The use of microdroplets in synthetic cell construction embodies principles of complexity science, where emergent properties arise from simple interactions and spatial constraints. As with biological systems, functions such as decision-making, adaptation, and self-replication can emerge from networks of interacting droplets. This modularity allows for the construction of increasingly complex synthetic systems from standardized, controllable units. Bridging the gap between chemistry, biology, and engineering, droplet networks with embedded logic gates have been created to mimic rudimentary computational behavior, such as responding differently to chemical inputs. It's like using droplets as programmable robots. Each one performs a task, and together they make up a synthetic organism capable of decision. Synthetic cells and microdroplets are far from equilibrium systems. They require continuous energy input via ATP, light, or chemical gradients to sustain active processes such as protein synthesis, membrane remodeling, or molecular transport. Thermodynamically, these systems transform external energy into ordered chemical transformations, mimicking the behavior of living cells while adhering to the second law of thermodynamics. The controlled confinement also enables gradient formation and localized energy storage. In a synthetic chloroplast model, light-driven reactions inside droplets produce ATP, which is then used to power internal enzymatic reactions. It's like a self-charging toy car inside a bubble. It runs only when exposed to light and uses its own little battery to perform tasks. Microdroplets provide an ideal environment for implementing synthetic gene circuits where DNA templates, enzymes, and small molecules process and generate information. Inside droplets, gene expression can be regulated with exquisite control, enabling artificial cells to sense stimuli, compute logic operations, and even evolve over time. Information is stored in nucleic acid sequences and interpreted by translation and regulatory networks, mimicking natural biological computation. A microdroplet containing a cell-free expression system was programmed to fluoresce only when two specific chemical inputs were present. Demonstrating AND-gate logic inside a synthetic cell, it's like a vending machine in a drop of water. It lights up and dispenses a response only if you insert the correct combination of tokens. Synthetic cells built from microdroplets hold immense promise in understanding the origin of life, designing minimal lifelike systems, and creating programmable biosensors, drug delivery platforms, and soft robots. By isolating specific components of living systems and reassembling them in droplets, scientists can dissect the minimal requirements for life and explore artificial evolution. These systems also offer templates for next-generation therapeutics and chemical factories operating in confined programmable units. Droplet-based protocells have been used as smart drug carriers that release their contents only in response to specific chemical signals present in diseased tissues. It's like a spy capsule that only opens when it hears the correct password, completely inert until the environment matches its programming. While powerful droplet-based synthetic cells face challenges including droplet fusion control, leakage, stability over time, and integration of complex functions, maintaining robust behavior under fluctuating environmental conditions, scaling production, and building complex communication networks among synthetic cells are ongoing research frontiers. Moreover, incorporating more lifelike behaviors such as autonomous growth, division, and evolution remains a technical hurdle. In some synthetic cell systems, lipid membranes become unstable after several hours, limiting their utility for long-term biological mimicry. It's like building a perfect miniature world in a soap bubble, but the world evaporates before it can grow or evolve unless carefully stabilized. The future of microdroplet-based synthetic cells lies in integrating advanced materials, microfluidic control, genetic circuits, and AI-assisted design to build fully programmable and responsive synthetic life systems. Such developments could lead to artificial life forms capable of sensing, computing, adapting, and interacting with real biological systems. These platforms will be vital for probing the origin of life, testing evolutionary hypotheses, and creating biohybrid machines with applications in medicine, sustainability, and robotics. Ongoing research aims to develop self-replicating droplet-based synthetic cells that autonomously grow, divide, and exchange genetic material, mimicking rudimentary reproduction. It's like building a sandbox universe where droplets evolve like creatures, learning, adapting, and perhaps one day outgrowing their test tubes.